Hi, good morning, everyone. I hope you're all doing well this morning. So today we are going to be doing lecture 3, which is just about cells. It's quite straightforward. And it shouldn't take us too long. And you know, I hope you guys found the quiz to be okay. Unfortunately, we're not able to show you your results. Like you can check your grades, but we cannot show you which question you got wrong because you know it's online learning and we have to maintain the integrity of the test bank. But you know, if you have any concerns or questions, then you can contact me privately, and you know, we will address that. Okay, somebody say I'm cutting off. Okay, let me see. Give me a second. Okay. Let me try to sign out and come back in and see if it's any better, okay? Hi, can you guys hear me? Hello, am I back on? Are you guys there? Yes. Okay. Am I still choppy or like it's acceptable right now? It's okay. Yeah. Okay. I'll keep talking for now. I switch to another channel, like another Wi-Fi connection. Hopefully this will be better. Okay. Let me know if I'm being cut off again. Okay. Then I might have to physically move to another location. Okay. So let's get started. Well, okay, in case you missed what I said, basically I cannot return your quiz to you for you to see which question you got wrong because we have to maintain the integrity of the test bank. But if you have any questions, concerns, like if you didn't think you'd do so well and you'd like to talk to me individually, feel free to contact me. Okay, let's get started. Lecture 3, cells. Okay, so stay 4 principles of the cell theory. Okay, so cell theory is one of those very important scientific breakthrough that advance us because we realize there are, you know, microscopic things that we cannot actually see, and by acknowledging that enables us to have breakthroughs in medicine. Right now, you know, we can study bacteria and know that they are the causing agent of various illnesses, right? So, you know, all that was possible because in part because of the cell theory. Okay, so let's take a look at what the cell theory is. First one is that every cell, every living thing or organism consists of one or more cells. Okay, so like if you think about humans, right? Humans, we have trillions of cells. Trillions of cells. Okay, and then if you think about like an amoeba, right? That's like one cell. Okay, so all organisms are made up of one cell or many cells. Okay. Another part of the cell theory is that a cell is the smallest unit of life. Okay. Individually alive as part of a multicellular organism. Okay, so like we have trillions of cells, but if you were to take some of my cheek cells, right? You can grow them in a petri dish, right? If you give them the correct nutrients and condition, you can grow them. Not indefinitely, like they're eventually gonna die, but you can grow them because individually they are alive. Okay, they don't really need to be part of the entire organism to live on, but you would need to provide support, of course, right? And that basically is what this second part of the principle says, right? The cell is the smallest unit of life. Life being something that exhibit all the characteristics we talked about in lecture 1, right? So, there's some complexity to it. There's going to be energy requirement. They have to maintain homeostasis. So all those characteristics that you will find in the entire organism, that you will also find on the cellular level, okay? Smallest unit of life. Another one is all living cells come from the division of pre-existing cells. Okay, sorry, it's getting a little bit crowded here. Of pre-existing cells. Okay, so we actually have a name for this, right? Anybody know what that is called? You might have learned it before. Cell division. What's another word for cell division?
Mitosis?
Mitosis. Thank you. That is correct. Okay, so no one really knows where the first cell came from. I mean, there are theories and stuff like that, and it depends, whether you believe in evolution or creationism. Nobody really knows for sure. Okay, but as far as we're concerned, all cells have to come from like a previous cell, okay, through cellular division. Okay, and then the last part is that cells contain some kind of hereditary material. Hereditary material. Now, it doesn't always have to be DNA, right? It could be DNA or RNA in some cases, right? But what's important is these hereditary material will be able to get passed on to the next generation as the cell divides. Okay, so these are the 4 principles of the so-called cell theory. Does anybody have any question? Okay. Next, we have fill in the blanks with the terms that correctly describe the organization of life from smallest to largest. Okay, so last class, we talked about atoms, right, which are basically everything on the periodic table. Those elements are made up of atoms. And when we combine atoms together, we form molecules, right? So small molecules like H2O, right, C6H12O6, those are important in our body. But then there are also the macromolecules that we talked about, right? The one that have thousands and thousands of atoms, right? So today we're going to move up one more. Oh, sorry, I should put macromolecule first. Macromolecule. Okay. So again, just to refresh your memory, this would be things like, just small things: water, glucose. Okay, and macromolecules, as you know, would be things like carbohydrates, right? Proteins, so on and so forth. And today's class, we will, move up at a level higher, and we're going to look at organelles. Okay, organelles are basically like tiny organs of the body, right? So if you watch the lecture recording, you should be able to give me some organelles, right? Anybody want to give me an organelle as an example? We don't have a lot of people here today, so like if you want to unmute and tell me, please, I welcome you to do so. Any organelles that you guys know of?
Cytoplasm.
Thank you, Nadia. Cytoplasm, it's what all the organelles are kind of bathed in. That the cytoplasm itself is not necessarily considered as an organelle. Anyone else want to try? Do you guys understand the question? Okay, so like you have your cell, right? Your cell, and then
Nucleus.
Nucleus. Sure, that is good, Yusuf. Thank you. So that is the nucleus, right? Okay, nucleus, right? And then there are a bunch of other organelles that we will look into more details now. But the fluid, right? The gel-like substance that all these organelles are kind of, bathing in, for lack of a better word, that's the cytoplasm. Okay, so cytoplasm itself is not really an organelle, but that's what all the organelles are submerged in. Okay, so example would be like nucleus. Very good. Okay, another one's a mitochondria, right? Endoplasmic reticulum, all these things that we talked about. So we will keep going higher and higher. So many organelles, you will find them inside a cell. Okay, inside a cell. So, example, we can say like a muscle cell, right? Muscle cell. It could be a brain cell, could be heart cell, liver cell, skin cell, what have you, right? And then multiple cells can come together and it will form tissue. So we have, many different types of tissues. We have 7 types of connective tissue in the body, for example. So we have things like, adipose tissue, right? So that's like fat tissue. Okay. There are epithelial tissue, right? So things that makes up your skin, the outer layer of your organs, right? So many different types of tissues, and tissues are basically a collection of cells, right, that come together to perform a specialized function. So adipose tissue, for example, the fat tissue is responsible for storing fat. Okay, and then you can have different types of tissue coming together to form an organ. Okay, so an example of an organ. Anybody want to give me an example of an organ here? Should be pretty straightforward. Any organs that you know? I'll put it here.
Liver.
The liver is good. Okay, so in the liver there are adipose tissue, right? There are epithelial tissue, as I said. There are fibrous connective tissues that holds them together, right? Blood is actually a type of tissue as well. So all these tissue come together to form an organ that would, perform a greater function, and that greater function would be for detoxifying your blood. Okay, so Caramel said kidney. That's another one. Very good. Now, but for the purpose of this flowchart, I am gonna use heart. Okay, not that there's anything wrong with liver or kidney, but I'm gonna use heart because the next level is gonna be the organ system. Okay, so the organ system is many different organs coming together to form a system that is vital for the organism's function. So the heart is part of the cardiovascular system. Okay, the kidney is part of the renal system. Okay, urinary system. Okay, the liver is kind of weird. It's part of the digestive system, but it's also part of your system that helps you detoxify toxins from your body. So it's a little bit tricky to place that one, but the heart would be part of the cardiovascular system. Okay, blood vessels would be part of your cardiovascular system. So in this way, we organize from the smallest to the largest. Okay, the largest being, of course, the organism. Okay, so make sure you know how to organize this. Right? You can bet there's going to be a question that asks you which of the following is the correct order. All right, next. List 3 common structures found in cells. Okay, so all cells are a little bit different, right? They look different. They might be found in different places. They might behave differently. But then there are three common features that you will have in all cells. Okay, so the first feature is they will all have a cell membrane of some sort. Cell membrane. Another word for cell membrane is plasma membrane. Same thing. Okay, cell membrane or plasma membrane. Both is okay. Right? Erase these quotation, you don't really need that. So, or plasma membrane. So that's the barrier that separates the outside from the inside, right? The non-living and the living on the inside. And actually, we will have a whole lesson on the plasma membrane next class. Okay. Another feature that is found in all living cell is that they all have the cytoplasm. Okay, as Nadia mentioned, cytoplasm, right? That's all the that's the gel-like substance where all the cellular content is floating in. Okay, and last, this is kind of similar to what you saw in the cell theory, is that they all have genetic material. Genetic material. Okay, and these are the things that gets passed on to the next generation. Now, generally speaking, we can separate cell types into what we call prokaryotic cells, right? The primitive, P for primitive, right? The more primitive prokaryotic cells, or the more complicated eukaryotic cells, okay? So the main difference are going to be summarized in this chart. Size, prokaryotic cells compared to eukaryotic cells, they are much smaller, okay? Smaller, and the eukaryotic cells are larger, okay? To give you an idea, okay? Prokaryotic cells are so small that sometimes they can invade a eukaryotic cell and live inside them, okay? So that's how the size difference would compare. If this is your eukaryotic cell, the prokaryotic cell might be this small. Okay. You guys heard of listeria before? Anybody heard of listeria? Sometimes you hear on the news, right? There's recall of, like, you know, dairy, deli products like turkey cuts, cold cuts, that kind of stuff. Sometimes they have listeria contamination. No, nobody heard of that? Maybe. Okay, so listeria is a bacteria, it's a prokaryote, and they actually will go inside the eukaryote and cause all kinds of problems. So the size difference. Next, structure, right? Not a lot of structures. Prokaryotes are quite simple. Okay, so they are simple, and the structure for eukaryotes are complex. Location of genetic material for the prokaryotic cells, they are just kind of, scattered in a central region. Central region of the cell. There is no specialized compartment for it, but for the eukaryotic cell, the genetic material is located inside the nucleus. So this thing here that you will always see is inside the nucleus. Now, I have to kind of give you a little bit of a heads up. What are we made up of? Just want to check to see if you're all with me. Are we made up of prokaryotic cells or eukaryotic cells? What do you guys think? What are we made up of?
Eukaryotic.
We are made up of eukaryotic cells. However, even though eukaryotic cells is supposed to have a nucleus, not all of our cells are going to have a nucleus. Okay, so some of our cells actually do not have nucleus. Does anybody have any example that you read about before, or just educated guess? Anybody know of any cells in our body that do not have a nucleus? I'm gonna zip my tea and wait for some response. Oh, interesting. A nail is actually proteins, right? As we learned last class. So that doesn't have any cell in it. But anyone else? Brain. We definitely need a nucleus in the brain cells because the nucleus controls a lot of processing, right? Power and stuff like that, and controls what the cell does. So we would want to have nucleus in the brain. So good guess, very good. Thank you for participating. Examples would be like red cells, okay? Red blood cells, right? Or things like platelets. Okay, platelets are the parts of the blood that helps you stop the bleeding if you're bleeding. So those cells, even though they're part of the eukaryotic organism, they actually do not have a nucleus, okay? Plural of nucleus is nuclei, okay? So some cells, they have more than one nuclei. Okay, I hope that makes sense. And again, if you have questions, please stop me, okay? And let me know. Okay, moving on. Number of organelles, few, okay? Just a few. And this one, eukaryotes, they have many. So examples of prokaryotic cells would be things like bacteria. Okay, and there are many different bacteria out there. Things, some of them that you might have heard of include things like E. coli, right? Okay, so I can put E.coli, right? Anybody know any other bacteria that they want to add? No? Okay, so people get strep throat, right? You might have heard of that, right? So that's caused by like staphylococcus, right? So that's another bacteria. Regardless, prokaryotic cells, an example of that would be bacteria. Eukaryotic cells are like, you know, animal cells, animal and plant cells. Okay, so like humans, right? A rose, right? Animal cells, plant cells. Yes, salmonella, Shannon said, yeah. So that's another example of bacteria. Very good. All right, salmonella. Yeah. If you ever touch like a reptile, if you touch like a turtle or like a lizard or whatever, they naturally have a salmonella on them, and you should always wash your hands. Okay. Next, we have labeled the different parts of the mitochondria. We'll talk more about this later, but mitochondria, that's a singular, and if it's a, then it's like plural, right? So what is the function of the mitochondria? Well, in very simple terms, the function of the mitochondria is to produce ATP, and ATP is basically energy for our cells. Okay, energy for our cells. So ATP is energy for our cells. Okay, so we need these mitochondria to make energy for us. So if you cut open a mitochondria, you will see that the inside is not hollow. I mean it's hollow, but then there are other stuff in it as well. Okay, so there is an outer layer, right, which we call the outer membrane. Okay, and then you can see there is an inner membrane here, and it's being folded up. So this is the inner membrane. Okay, maybe it's a little bit hard to see, so let me try to color it, outline it for you. Let's use green. You see, this is the inner membrane, right? Inner membrane. Okay, and I'm not going to go all the way up. You know what? Let's just go all the way around. Okay, so the inner membrane is actually all folded up. Okay, so it folds into finger-like projections. Sorry, can't spell today. Projections. Finger-like projections called cristae. Okay, and you want to know these parts because when we do metabolism in lecture 5, I believe, we will have to know the parts of the mitochondria quite well. All right, and here, right, the outer membrane, it's just this part right here, the outer membrane. So there would be some space between the outer membrane and the inner membrane. So that space is called the intermembrane space. Intermembrane space. And then inside the inner membrane, all this area right here, right? That's filled with another gel-like substance, and that is called a matrix. Okay, so there are different functions associated with these various parts, and we will talk more about it when we get to the metabolism unit. Any questions so far? All right, let's move on to talking about the different organelles, and we will describe them and we will talk about their functions. Okay, so first one, what is this pointing at? Okay, by the way, this is like everything in this chart is part of the endomembrane system, right? Okay, so the endomembrane system is basically a network of membranes that are found within the cell. Okay, so let me write that down for you. A network of membranes inside the cell. Okay, endo means within, right? Okay, so we have the cell membrane, which surrounds the entire cell, and inside there are a bunch of other organelles that are also made up of a bunch of membranes, and we call them the endomembrane system. Okay, so they have interconnected functions. Okay, so we will start with the first one here. Okay, so this one is pointing at, I mean, there are two things shown here, and we are pointing at, you know, just this part right here. Okay, not down here. So this part right here, that is the rough endoplasmic reticulum. Okay, or RER for short. Okay, now on eCentennial, there is a link that takes you to like a 3D cell model. I mean, it's not the best one out there, but it's pretty simple. Okay, the other one that looks better are very complicated. Okay, so here you can see this is the cell, right? And you have the nucleus in the center, and around the nucleus you will see there are all these membranes, right? Okay, so these purple ones here, that's the rough endoplasmic reticulum that we are talking about. Okay, and then further out, these little, you know, beige color sausage looking things, those are the smooth endoplasmic reticulum. Okay, so nucleus, and then outside of that you have the rough ER, and then further out you have the smooth ER. Okay, so I encourage you to play around with this. You will find it in the extra resources under the lecture 3 folder. Okay, so what does it look like, and you know, what does it do? So I will do description in red, and then I will do like function in blue. Okay, so description, right? Well, it would be part of the endomembrane system. Part of the endomembrane system, and it is actually continuous. That means connected, right? With the nuclear envelope. Okay, so we haven't really talked about the nuclear envelope up to this point yet. I mean, I talked about it in the lecture recordings, but basically, very simply, you have your nucleus. The nucleus is in the center where the genetic material is found, and the nucleus is surrounded by the nuclear envelope. So that envelope continue to extend outward, and it will form the rough ER. In the 3D model here, right? This purple layer that has all the pores on it, that's the nuclear envelope, and it will be fused together with the rough ER. Okay, so I think if I click on it, it will show you that this is the nucleus with the nuclear pores, and you know, it doesn't really call it the. Oops, sorry, this is not really working well, but it's sorry. That here, it's the nuclear envelope. Okay, and again, it's continuous with the rough ER. Okay, so that's the description. And also, it is going to be covered with ribosomes. Okay, so ribosomes is another organelles that we're going to talk about. And their function is to make proteins. Okay, so that's the description. Okay, let's do function. Now, rather than like cramming it all in here, you probably will have more space, okay? Because then your pencil is probably thinner than my marker here. But I'll put the functions here on the side. Okay, so what does it do? Well, what it does is that the ribosomes that are found on the rough ER will synthesize proteins directly into the inside of the rough ER. Okay. So let me try to draw this for you to show you. So this is the rough ER. Okay, something like this. Okay. And on it, you will find a bunch of ribosomes stuck to it. Okay, these ribosomes. So with all these ribosomes on them, it makes them look bumpy, makes them look rough under the microscope. Okay, so what happens is the ribosomes will now produce a protein directly into the inside of the endoplasmic reticulum. Okay, and what happens is the protein, as you know from last class, they will fold up right into a 3D shape, and when it's all ready, it will move to the edge and pinch off in a little vesicle. So now the protein is inside like that. Okay. So another function is proteins will be packaged into vesicles that gets pinched off from the rough ER. Okay, so that's basically the function of the rough ER. Okay, so you would expect cells that makes a lot of proteins to have a lot of ribosomes. Okay, so cells that makes a lot of proteins would have a lot of ribosomes. Okay, maybe I'll put it here. Right. Cells that makes lots of proteins will have lots of rough ER. Okay, so example of a cell that makes a lot of proteins would be a cell in the pancreas. Okay, so last time I think I gave you a poll question, right? The pancreas makes insulin, right? Insulin is a protein, so you would expect the cells there to have a lot of rough ER. Okay. Next, we have the smooth ER. Okay, so smooth ER is a little bit further out from the nucleus, and it's smooth because it does not have any ribosomes on them. Okay, so this is the smooth endoplasmic reticulum. Okay, so also part of the endomembrane system, right? Part of the endomembrane system. They all are. So really, I don't really have to write this every time for this chart, but let's just do. Okay, and they are not covered with ribosome. Okay. The function. Okay, I think you'll be able to fit it this time. The function is that they are important for lipid synthesis. Okay, they will produce the lipids, and some important lipids that we talked about last class include things like testosterone, right? Estrogen. These steroid hormones, they are made in the smooth endoplasmic reticulum. Okay, and also, in addition to making lipids, the smooth ER is also responsible for detoxifying. And breaking down lipids. Some lipids we don't want it to accumulate in the body, and drugs. Okay. So which organ is responsible for metabolizing drugs? Does anybody know? You ever heard that like if you take too many Tylenols too often, which part of the organ is going to be affected? Yeah, exactly, right? Someone said
The liver?
the liver, exactly, right? So liver. So you would expect to have lots of smooth ER in the liver, right? Make sense? Okay, moving on. Is everybody okay? Like, I know I'm not really asking a lot of questions, but like you guys seem very quiet. I just want to make sure you guys don't have any questions. Okay, perfect. I know this is not the most exciting thing. I promise you, next semester will be very exciting when you learn about the human body. Lots of fun and exciting things. But this is the foundation for that. Moving on, okay. We have these things that kind of stacks up like pancakes. We call them the Goji body. Goji body or Goji apparatus, okay? Or Goji apparatus. Doesn't matter what name you use, okay? I'm just showing you in case you read about it. So again, right? Part of the endomembrane system, and it's going to be instead of being like continuous channels, membranes like the ERs, these ones are like individual, okay, separate stack. So if I have to draw it, it would be like one stack here, 2, 3, 4. That would be the Goji, okay, as opposed to the ER is more like, you know, continuous. You guys understand? Okay, so separate versus continuous. Okay, so I'm going to erase that so I will have space. So that is layers of membranes stacked together. Okay. They might not be physically touching each other. They're very close to each other. Okay, so what does it do? Well, the best way to think of this would be like post office. Okay, so what they do is they will sort, okay, and sometimes modify, and repackage vesicles, proteins that arrive in vesicle. Okay, so just let me go back here for a second. Remember this one over here, this guy that gets pinched off from the rough ER? Well, where is it going to go? Well, some of it is going to go to the Goji body. Okay, so over here, it's going to go to the Goji body. Right, so this one might come in and it will fuse with the first layer of the entire Goji body stack. And then the content will get sorted, modified. Okay. In fact, this will actually get passed on forward and sometimes backwards a couple of times. Okay, back and forth a couple of times until it's finally ready. Then it will get, sent off in a new vesicle. Okay? Sometimes you don't have to modify it. Sometimes it will just, get sent off immediately almost. Okay, but like it needs to check. And how does it check? Well, it's a complicated process. Okay, the polypeptide protein actually have a few amino acids at the end that serves at like almost like a barcode, like a scanning tag. Okay, so based on what kind of tag you have, then the Goji body will know where this needs to go, or if it needs to be modified, right? You need to add like sugar to it. You need to add like other things to it. Then that little signal is what will tell the cell what needs to be done. Okay, so it will get packaged, modified, and sorted. Okay, so after it's done, then some of the vesicles, it has basically like 3 destinations. Okay, so the vesicles that gets pinched off from the Goji body. Okay, number 1, it would either move to the surface of the cell and it will get released. Okay, so let's say this is a pancreas cell and it's making insulin, right? So once the insulin is made, it will get released out of the cell into the bloodstream. So that's one destination. Number 2, right? It will get repackaged into a vesicle, and the vesicle will be stored in the cytoplasm until you need to use it. Okay, so maybe you are not eating a meal right now, so you don't need the insulin to be released immediately. So you will store them in little vesicles, and then we will release it later on. Okay, or in some cases, it will become like another specialized organelle that would carry another specific function. So this one is called the lysosome. Okay. So also part of the endomembrane system. You must be sick of writing this line now. Part of the endomembrane system. Okay. And it is a vesicle that contains various enzymes. Okay, so lots and lots of enzymes. What does it do? Well, a lot of things actually. It will digest old organelles. So if there is like mitochondria that's not functioning anymore, then the lysosome will fuse with it and then break it down, and then in terms release some of the resources to be reused. Okay, that's one possible function. Digest old organelles, or it could break down break down molecules, or sometimes bacteria ingested by the cell. Okay, so an example of this would be white blood cells, right? White blood cell. Okay, so you have many different types of white blood cells, and I will teach you all the glory details next semester. But one of the white blood cells that we have is this thing called like macrophage. Okay, macro means big, phage means eater. So this is literally the big eater. I'm not making this up. This is for real. Okay. So what does the macrophage do is the macrophage is like Pac-Man. If they see a bacteria, right, then it will eat it. Okay, it will eat it. Okay, so like inside the macrophage, there will be a lot of lysosomes. Okay, so let me just draw one lysosome here. Lysosome. Okay, and this would be like the nucleus. Okay, so what happens is the macrophage will eat the bacteria. Okay, it will basically like surrounds it like that. Okay, and then afterwards it will this part here will join with this part, and then the bacteria will be trapped inside like this. Okay, so that's again the lysosome here, and what happens is the lysosome will now fuse with this vesicle that has the bacteria in it. And then all the enzymes will now attack the bacteria and break it down. Okay, so that's one example of what the lysosome is for. Does anybody know what this process is called? That the macrophage is like eating the bacteria. Like if you took biology in high school, do you remember what it is called? Starts with a P. Starts with a P. Anybody?
Phagocytosis.
Thank you, Nadia. Phagocytosis. Okay. And if you've never heard of this word before, that's okay. You will learn it next lecture. Okay, that's that. Now, in the lecture recording, I also talk about there are some lysosomal diseases, right? So let's just quickly review those. So lysosomal diseases. Okay, so that's when, like I said, there are many enzymes in the lysosomes, right? And what macromolecule makes up the enzyme? That's a question. What macromolecules makes up the enzyme? Is it carbohydrate? Is it lipid? Is it protein? Or is it nucleic acid? Which of those macromolecules makes up the enzyme? You know what? Let's do a poll. Since you guys are not really answering. Okay, so my question, what are the enzyme made up of? Is it A, carbohydrate, B, protein, C, lipids, D, nucleic acid? What do you think? What are enzymes made up of? Okay, only 4 of you, 5 of you have replied, 6 now. Okay, I'll wait a couple more seconds. Okay, so the correct answer is proteins. Okay, proteins. Enzymes are proteins. Okay, which means they are encoded by the DNA. So if the DNA has a mutation, then the protein will be misfolded, right? We talked about this last class. Okay, so there are a whole list of genetic conditions where the enzymes in the lysosomes are non-functional. Okay, so depending on which enzyme is non-functional, the manifestation of the disease will be different, right? So I believe I talked about 2 diseases in the lecture. So there is the Pompe disease. Pompe's disease. Okay, so this one, what happens is the enzyme, one of the enzyme is mutated, and it leads to the inability to break down glycogen in the muscle. Okay, so glycogen is a storage form of energy in muscle, right? As you know, and liver. And what happens is if you can't break it down, then they start to accumulate. Okay, and too much of a good thing is not always going to be ideal. Right? So what happens is this leads to toxic accumulation of glycogen in muscle. Okay, especially the heart. Okay, and that's going to cause some cardiovascular conditions. Okay, and then there's another one. Okay, I'm just going to write over this picture. I'm sorry. There's the Tay-Sachs, right? Tay-Sachs disease. Right? And this is the toxic accumulation of lipids in brain cells. Okay, and that could lead to developmental problems. Okay, in fact, if a woman is pregnant, she is at risk of carrying a child with Tay-Sachs disease because of family history and other genetic factors. She could do what's called amniocentesis. Has anybody heard of that before? Amniocentesis? Anyone want to tell me what that is, or not sure what that is? Yeah, Shannon, go ahead.
An amniocentesis is when they draw a small amount of the amniotic fluid that surrounds the fetus to test it for genetic abnormalities.
Excellent. I could not have explained it better. Okay, so the baby is surrounded by these fluids, right? When the parent, when the mother is about to give birth, you hear the phrase like the water breaks, right? So that water is actually the amniotic fluid, and it contains some of the cells from the baby. So what you can do is at I think around 21 weeks is safe. There are risks, but it's relatively safe to do so, to insert a needle, right, guided by ultrasound, to take out some of the fluid from the mother, and then analyze the DNA from the baby cells that is in that fluid, and you can screen for a whole list of genetic conditions. Okay, things like Down syndrome is one of them, trisomy 18, and then here is the Tay-Sachs disease that you can also screen for. Okay, so that is the endomembrane system, everybody. Starting with the rough ER, smooth ER, Goji, and lysosome. Okay, so I ask again, any concerns, any questions before I move on? All right, fantastic. Next, list 3 organelles or structures that are unique in plant cells. Briefly describe their functions. Okay, so can you type in the chat what 3 organelles or structures that are only found in plant cells but not in animal cells? Okay, please go ahead and type your answer. It's okay if you are guessing, that's all right as well. That's part of the learning. Okay, you don't have to get it right necessarily, but please participate. Is it because some of you don't know where the chat box button is? Is that part of the problem? Let me do a quick poll and see what the problem is. If you know where the chat button is, please choose A. If you're not sure, please choose B. The chat box. Oh, okay. Looks like most of you know where the chat box is. You just don't feel like talking, right? That's okay as well, I guess. That's okay as well. Okay. But yes, some of you are typing now in the chat box. Shape of cell membrane. Okay, interesting. Chloroplast. Yeah, chloroplast is a good one. Anyone else besides chloroplast? Let me write that down. Chloroplast. Okay, so wall, yeah, or more correctly, cell wall. Okay, but now compared to animal cells, we don't have cell wall. Okay, but like bacteria, for example, they could have a cell wall. And there's one more that I'm looking for. Anyone else? Anyone else know what the last one is? Okay, I'll do the last one. It's called the central vacuole. Okay, central vacuole. Okay, so what is the chloroplast? Chloroplast is a double membrane organelle, just like the mitochondria. Okay, there's outer membrane, there's an inner membrane, right? And you know, instead of having the matrix, it has something called the stroma, but we're not going to go into it. It's a double membrane organelle, and it contains, very importantly, embedded within these the membranes is the chlorophyll. Contains chlorophyll. Okay, which enables photosynthesis. Okay, so photosynthesis is to use the energy from light to convert carbon dioxide into sugar. Okay, so that's how plants make their food. We cannot make our own food. We have to consume the food, right? We have to eat it. But plants, they can do this. Okay, so again, you know, in this question, I should say list 3 organelles or structure that are unique in plants. You know, compared to animal cells. Okay, because you actually will find chloroplasts in some other cells, okay, that are not animal cells. Right? There are some cells like protist, right? That's what amoeba belongs to. So some of these organisms that lives in pond or lake, they sometimes have chloroplasts as well. Okay, so it's unique to the plant compared to animal cells, but not like entirely unique. Same thing for the cell wall, right? Okay, so cell wall is made up of cellulose, and what we learned last time. Okay, it protects the cell from bursting. Okay, when there's too much water. Okay, also provides structural support. Okay, and then there is the central vacuole. Okay, so the central vacuole takes up a lot of space in the cell, takes up more than half of the cell's volume. Okay, what it does is it stores mostly water and other nutrients. Okay, in some cases, it can also store pigments, so like colors for the flower petals, for example, or poison in some cases, or poisons for protecting the plants. Okay, so those are the three things. Any question? Okay, so why don't we take a quick break, just maybe 5 minutes, okay, so you guys can recharge a little bit, and we will resume at 9:35. Okay. Okay, let's continue. But before that, maybe we will do a quick poll question. Let's see where this is. Okay. Go ahead and do that, please. Okay, sorry, my kid's school is calling. I have to answer this. I'll be right back, okay? Sorry. Okay, sorry about that, everybody. So yes, the correct answer is D, right? I said it's a basic structural and functional unit of living organism, okay? So that is that. Let's try another one, okay, please. Okay, go ahead. Okay, a couple more seconds just to get a few more results in. Okay, so we learned about the endomembrane system. I told you it's possible for it to move to the cell membrane, it get released, and sometimes they actually can become part of the cell membrane. It definitely can be inside the lysosome, right? Those enzymes that we talked about are an example of that. They could be floating in the cytoplasm and waiting to be used at a later time, but they will not be in the smooth ER. Okay, smooth ER is not responsible for producing proteins. The smooth ER is where you will be producing lipids. Okay, so the answer would be C. Okay, you will expect to find the proteins in A, B, and D, but not C. Okay, I hope that makes sense. Let's continue with this thing. Okay, so these are extra organelles, and they are not part of the endomembrane system anymore. Okay, so here you can see this is a nucleus. Okay, so what is the description of the nucleus? Well, for nucleus, it is found in the eukaryotic cells, and it's covered by the nuclear envelope. Okay. There are nuclear pores on the nuclear envelope. Okay, so all these little bumpy dots that you see, they're actually pores, they're little holes for things to go in and out of the nucleus. And I'll tell you what goes in and what comes out in a bit. And this contains two things. It contains the chromatin and something called nucleolus. Okay, so a lot to unpack there. Let's talk about it. Okay. So first of all, let's talk about the nucleolus. Okay, the nucleolus. Nucleolus is responsible for synthesizing. Synthesis of ribosomes. Okay, remember those ribosomes that are covering the rough ER? Well, where do they come from? Well, they came from the nucleolus, which is like this part here. If you think about like a peach, right? The seed, the big core, the seed, right? Inside a peach, that would be the nucleolus. Okay. So the ribosome will exit through ribosomes will exit the nucleus through the nuclear pores. All right. Next, we have the chromatin. Okay, so they are like found all over here, right? So you probably have heard of chromosome, okay, like from, basically everywhere. So what's chromosome and what's chromatin? Well, to understand chromatin, you should understand what chromosome is first. Okay, so you've probably seen chromosome drawn like this. Okay, so chromosomes are basically chromosome. They are condensed DNA fibers. Okay, so think of like sewing threads. Okay, so sewing threads is DNA, but when you crumble it together, make a ball, it condensing it, right? Then it's much more visible compared to a single thread, right? Essentially, that's what chromosome is. Chromosome, it's like when you take the DNA fiber and you condense it a lot, then it becomes chromosome. And when it's really condensed, it's easier to move them around without tangling them. And you would want to move the chromosomes around during cell division. Okay, so normally when a cell is not dividing, then the chromosomes the DNA will not be forming the chromosomes. Okay, so they are rather invisible under the microscope. Right? It's only when the cell is ready to divide, then the DNA condenses itself into the chromosome. Okay, so what does the DNA look like for the rest of the time? Well, for the rest of the time, it exists in the form of chromatin. Okay, so chromatin is basically DNA plus packaging proteins. Okay, so not the condensed version, not the chromosome. Okay, this is just like a more relaxed version, a more opened up version. Okay, and you want that. You don't want it to be super compact. When it's more open, you can access the genes easier. Okay, so that's the difference. Okay, and then I don't remember if I'll talk about this in the lecture, but there's something called the central dogma of biology. Okay, a dogma is like a theme. Okay, So what's the central theme in biology? Well, you start with DNA, which contains the instruction to make the proteins. And then through a process called transcription, you're going to make a copy of some of the DNA. And that copy is going to be in the form of RNA. And then you're going to use that RNA to make proteins. Okay, so this is called transcription. And this is called translation. So in order for this to happen, once the DNA is copied into RNA, the RNA will now exit into the cytoplasm through the pores. Okay. So over here, the nuclear pores, right? Okay. So both RNA and ribosome passes through the pores. Okay. That's it. A lot of stuff for the nucleus. Next, we have these ribosomes. Okay, so ribosomes, they are very, very small. Okay, very small compared to the other organelles, right? They might be free flowing in the cytoplasm or stuck to rough ER. We know that already. Okay. It also contains a large and a small subunit. I should make these boxes bigger for next time. I keep on forgetting that. So like if you zoom in a lot, right, it actually looks like a hamburger or like a macaroon, whatever you like to eat. So there's the large subunit on top, and then there's the small subunit at the bottom. Okay, and then the RNA that I was talking about earlier will be sandwiched between it. And then the RNA will be decoded by the ribosome. And the main function of the ribosome is to make proteins. Okay, so some proteins are made freely in the cytoplasm. Some proteins are made in the endoplasmic reticulum and get packaged into little vesicles. One other thing I should mention, I think it's also in the lecture note, is that the ribosome is made up of RNA and protein. Okay, so the ribosome makes protein, but it itself is also made up of proteins and a little bit of RNA as well. Okay, so that is that. Next, we have the mitochondria. Okay, so ION for singular, IA for plural. So we talked about the structure already, right? It is going to have the double membrane, right? So see the diagram above for more description, okay? But what is it for, right? This is the energy production site. Okay, so produces ATP through a process called cellular respiration. Cellular respiration. Okay, so that's like glucose plus oxygen oxygen produces carbon dioxide plus water and up to 36 ATP. Okay, so that is the equation. And we will learn about this in a lot more detail later on. Here, the next thing, we have the centrioles. So they are, sorry, I forgot. There are a couple other additional information I should tell you about the mitochondria. I'll write here. And again, I'm sorry, it's like so crowded. The mitochondria have their own DNA for encoding some of their own proteins. Okay, did you know all your mitochondria came from your mother? Right? When the egg and the sperm fuse together, right? The sperm only injects the genetic material. The mitochondria from the mother's egg is what gets passed on. So all your mitochondrial DNA is inherited through the mother. If there are some mutation in this DNA, then it could lead to mitochondrial diseases, okay, as opposed to lysosomal diseases, right? So mitochondrial diseases, depending on which gene is mutated, can be related to many different outcomes, okay? Cardiovascular problems, high blood pressure, diabetes, to name a few. Okay, moving on. Excuse me. Centrioles. Okay, so what are the centrioles? Usually they will exist as a pair and arranged perpendicularly, 90 degrees to each other. Okay, and the function is they will produce, okay, sorry, produce spindle fibers during mitosis to guide chromosome movement. Okay. Next, we have the cilia. And cilia, basically, you would, if they exist, they would cover the entire cell surface. Okay. So either they help cell movement, okay, or move things around the cell. So what's the difference between the two? For example, there is something that looks like this, okay, with a mouth here, and then it has a lot of cilia around it, okay. So this thing is called like a paramecium, okay, never mind the name, okay, but it's a single cell organism and it lives in pond water. So to move around, it would use these cilia to pedal, and then it's, moves around in the water. So that's like cell movement. In our body, we have cilia in the respiratory tract, for example, okay? So in our respiratory tract, right, the cells, they would have, microscopic cilia. So if there are some dust particle trapped here, or if there are bacteria trapped here, then the cilia will sweep them up to your throat where you can spit it out. Okay, so all that cilia movement is not to move the cell, but rather to move things around the cell. Okay. Next one is the flagellum. Okay, and the flagellum, usually just one, per cell. In humans, definitely just one. Okay, like if there's a sperm with two tails, you can search it up on YouTube. amazing what you can find on YouTube. Okay, but like if the sperm with two tails, instead of swimming forward, it just goes around in circle. It doesn't go anywhere. Okay, so one tail, okay, one flagellum per cell. Sometimes two in some organisms. Okay, so there is something called like a euglena. It's like part plant, part animal cell, and then it has two tails. Euglena, I think that's how you spell it. Okay. And then, of course, the function is cell movement. Okay. Any question before we continue? All good? All right. So next we have to fill in this chart. And I think that is the pretty straightforward part. Directly involved in protein synthesis. Okay, so based on what I taught you, you should know this is the ribosome. Okay. The region inside the cell, except for the nucleus. That would be the cytoplasm. Organelle that manages or controls all the cell functions in a eukaryotic cell, that would be the nucleus. Digest excess or worn out cell parts, food particles, or invading virus or bacteria, that would be the lysosome. Small bumps located on portions of the endoplasmic reticulum, those would be the ribosomes, which again is needed for protein synthesis. Involved in detoxifying harmful substances and digestion of fat, and produces hydrogen peroxide, which is H2O2, as a byproduct. Okay, so this one is particularly like the hint here is the hydrogen peroxide, right? So it's something called peroxisome. Okay, it's similar to lysosome, but it's not part of the endomembrane system. Okay, this one uses oxidation reactions to detoxify things. Okay, also a lot of peroxisomes in livers, particularly when you break down alcohol and stuff like that, that's the peroxisomes. Okay, so H2O2 hydrogen peroxide is generated as a byproduct. You cannot let the H2O2 out of the peroxisomes. Okay, it needs to be broken down into water and oxygen inside the peroxisome. If the H2O2 gets out, it's toxic. It could damage. It could do a lot of damages. Okay, so oxidation reactions are reactions that involves removing electrons from things. So that is peroxisomes. Produce a usable form of energy for cell. That is the mitochondria. Sort and modify contents. Goji body. Site where ribosomes are produced. That will be inside the nucleus in something called a nucleolus. Provides support for cells. So cytoskeleton. Okay. There are three types of cytoskeleton, and I talked about it in the lecture, but very briefly, there is the micro filament. There is the intermediate filament. And we have the microtubule. Okay. So microfilaments are made up of something called actin. You have a lot of that in the muscle. It helps you, produce muscle movement, for example. Intermediate filament helps you provides structural support. It locks two cells together, for example. Maintains cellular integrity. Microtubules, they form these tracks for vesicles to move through. Like, how does the vesicle know how to go from the, endoplasmic reticulum to the Goji body, for example? Well, there is like a microtubule tracks that would guide the vesicles to its destination. Okay, so that would be cytoskeleton. Small hair structure used for movement, that would be cilia. Right? Usually you have more than one. Singular of cilium is cilia is cilium. Form a selective permeable barrier around the cell. That is the plasma membrane. Long whip-like structures used for movement. Flagellum. Contains chlorophyll needed for photosynthesis. That's chloroplast. Large sac filled with water in plants. Central vacuole. And finally, produces microtubule fibers needed for guiding chromosomes. That is the centrioles. Okay, so the spindle fibers is actually made up of microtubules, right? Remember, guiding system, these tracks that would guide the movement of the chromosomes. Okay. So that is that. Now, long answer questions, as well as application questions. Does anybody have any requests? There are only 3 questions here. I think they're relatively self-explanatory, but does anyone want me to take up any of these in particular? Only 3 questions. Are we okay with that? Okay, I will take that as a yes, you're okay. Let's talk about the application questions. Number 1, speculate what will happen to an animal cell if it is placed in pure water. Do you expect the outcome to be different for an animal cell? So that is actually something we'll talk about next class, but I want you to think about this, at this moment, right, before we learn about it. So if we put animal cells in pure water, so let's say this is a beaker and, it's just water there, and then we put the animal cell. Okay, so there are so many things inside the animal cells, right? There are some nutrients, there are these organelles, there's proteins. So what happens is if you put it in pure water, then the water will try to go into the cell, right, to dilute the content. This is called osmosis. And again, you will learn it in more detail next class. And as a result, the animal cell will swell and actually burst, okay, and die. Plants, on the other hand, is not gonna have this problem, okay, because plant cells, they would have the cell wall. So when the water goes in, right, the central vacuole will absorb it all, and the cell wall will protect the cell from bursting. In fact, plants, as you know, you got to water your plants, right? Otherwise they wither. So that water pressure will help prop up the plant. Okay, so the cell, a plant cell will not burst. Okay, in fact, it will be very happy when this happens. Okay, next one. Your heart muscle needs to function continuously throughout life. Which organelle would you expect to be most abundant of in the heart cell? So what organelles do you think you will find a lot of in the heart? Anybody? Of all the organelles that we talked about, your heart needs to beat all the time. What organelle would you expect to be most abundant? Marisha said mitochondria. That's correct. Okay. And it's not too difficult to justify because mitochondria produces ATP, and that's what you need, right, for those continuous contractions. Next one, endocrine glands produces and secrete hormones. Which organelle would you expect to be most abundant in cells of endocrine gland? So this is a little bit tricky. There are a couple answers here. I will see if anyone wants to answer. Okay, Maria said SMOVER. Okay, yeah, that's part of the answer. Okay, so depends. Okay, if the gland produces protein hormones, then it will have lots of rough ER and probably Goji body. Right? If the gland produces lipid hormones, then it will have lots of smooth ER, right? Because lipid synthesis occurs in the smooth ER, right? Whereas protein synthesis happens in the rough ER. Okay. All right, and just one last poll question before I send you off. This one is multiple answers. So please choose all the appropriate choices. Okay. So select all false statements. So let's take a look. The first one, smooth ER is part of the endomembrane system. We know that is correct, so that's not the answer. Muscle cells are expected to be abundant in mitochondria. It's correct, right? You would expect to have a lot of energy producing mitochondria in muscle cells. So that's not the answer. There are multiple nuclei in single brain cells because high processing power is needed. That is, it sounds reasonable, but actually brain cells only have one nucleus, okay? All cells would only have one nucleus. All cells have cell wall to protect itself from the environment. No, animal cells don't. So C and D are the false statement. Okay. Well, that's it for today. Next class, we'll do lecture 4, and your quiz number 2 is going to happen in 2 weeks, just before the engagement week. Okay, so for those of you who don't know what engagement week is, it's basically like, you know, what they call like reading week. Okay, it's a week where you don't have you know, lectures. You know, the college provides various activities for you to join if you're interested in, and you know, you can kind of think of it as like a break from school. Okay, and then we will come back and do lecture 5, which is the last lecture of the unit, and then we're going to have a unit test at the end of October. Okay, but for now, right, next week we will do the synchronous lesson on Friday, and then we're gonna have the lab and the quiz in the following week. So no, yeah, we won't have any assignment for the engagement week. The general consensus in the college is that's a week for you to take a break. So typically people don't assign any work or a quiz or test for the engagement week. Okay, so you will hand in your lab before you go on your break, and then you don't have to worry about it. And in fact, there's no test or quiz the week after for my course. So, you know, you can burn your notes if you want. Okay. Other than that, I will see you all next week, and please email me if you have any questions. Okay. Have a wonderful weekend, guys. Bye for now.
Thank you, sir. Bye.
Bye.